All right. Well, good morning, good afternoon, good evening, wherever you may be. I know people are still trickling in, but it's 2 minutes after the hour, so we're going to go ahead and get started. Really excited to present on this webinar on this topic, the role of microbiomes in modern soilless cultivation. It's a topic that Jiffy and myself are really passionate about, and we've been trying to dive further into this topic and have a great lineup of information and speakers to share with you. So to just a couple of housekeeping announcements before I share our speakers. In Teams, you should see a Q&A function across your control bar. You can use that to submit questions throughout the webinar. We will take questions at the end of the webinar, so we will all go through our segments and then we'll address Q&A at the end. But please use Q&A to add those questions. The chat is enabled, so if you want to add any comments or anecdotes or chat with anyone on the webinar, that's totally fine, but let's try and keep questions through the Q&A. We will be recording this, so we'll be able to provide this after the webinar in case you need to go back and check something or interested in sharing it, so we'll be able to provide that to you. My name is Kyle Freedman. I'm the Global Segment Manager for Controlled Environment Ag at Jiffy Group. We're one of the leading horticultural growing media companies in the world and excited to talk more about microbiomes in CEA. I'm joined by Paul Rutten, who's the founder and CEO at Concert Bio, and I've been collaborating with Paul for a little over a year on this topic and trying to shed more light on it. And we are also joined by Marta Stremińska from Wageninegn, who is a microbial ecologist. So we have some heavy hitters on this call, not myself, but Paul and Marta, who know a lot about microbiomes, and so some great information to share. We did publish a white paper this past winter that provides more detailed information on this topic. That was a collaboration between Jiffy and Concert Bio. Grateful to have Wageninegn from the research side in this webinar, but we'll be able to share the link to download the white paper following the webinar if you have not downloaded it already. So just to frame ourselves a little bit with challenges in soil cultivation. And so we're going to talk a lot about shifting from soil cultivation to soilless, and many of us who are within the controlled environment ag space or indoor ag know this and have experienced this, but there's a lot of different areas that we've seen that have contributed to this shift, whether it's soil degradation or having a lack of arable land for conventional outdoor farming, soil-borne pathogens, or just pests and disease in general. That pressure is a lot greater, we know, in outdoor production systems. Water management, so precision irrigation management is a little bit more difficult outdoors and access to water. And then climate change and erratic weather is a huge reason why some of this shift to indoor cultivation has happened. When we talk about the benefits of soilless substrates and soilless cultivation, they're numerous, but there's a lot of waste and sustainable materials that we can use to create soilless substrates. So the amount of materials that we can use are really endless. Clean and disease-free, there's a lot of effort to engineer these materials, process them to be pathogen-free. Reduced water use, and I think in general, just precision irrigation and nutrient management is a reason why we've shifted to soilless cultivation. And then the fact that you can use soilless substrate and create these systems in low to high tech environments is a huge benefit. So it's extremely flexible and location agnostic. So soilless cultivation can happen anywhere with any type of system and technology. But when we move from soil to soilless, we know we're working with these outdoor environments and we're moving into more complex, highly technical, automated environments. And part of what we've done is to try and take what we know about outdoor cultivation to improve root zone environment, to modify our environmental factors to really enhance production, and then limit pest and pathogen presence and disease. But I think a big issue and lack of understanding that we have within our industry is that when we go from soil to soilless, we're a little bit throwing the baby out with the bathwater because we're trying to take all of the elements that we know about production, control them to enhance cultivation, and eliminate all of the pathogen pest presence, but we're also limiting some of the positive microbial elements that we see in outdoor environments. And so this dichotomy of trying to create indoor environments that are completely sterile, completely controlled, and without this microbial diversity is a driving reason why we're interested in looking at this topic further. When we look at principles of soil health, this is a schematic I pulled from the U.S. Department of Ag's NRCS. Many of us know with outdoor conventional production that improving soil health is extremely important. The science and data behind soil health has grown over the years as we've learned more about it, as more research has been conducted. And some of these principles, minimizing disturbance, maximizing soil cover, we know that to be true of outdoor. But then there's two main facets of maximizing biodiversity, maximizing living roots, and that's all contributed to these topics and research areas of building soil health, living soils, regenerative ag, that now we have the data and years of research to know that soil health and making sure that there is a diverse living environment within our soil is really important, not only for resilience of production systems, but increasing growth and development of plants. When we talk about principles of substrate health or substrate quality, a lot of times we focus on these physical and chemical elements. And so, as I mentioned, soilless substrates are highly engineered materials that we can manipulate and change based on the system, based on the environment, based on the crop, and that can create a really healthy and quality substrate. But I think we can sort of extend this to not just be physical and chemical components, but I think we also have to better understand what are the microbial elements of substrates that we should be aware of that we can leverage. When we look at microbial diversity, this is some data that came from Concert Bio, and Paul is going to talk more about this in detail. But I think it's very obvious that we know soil-based systems outdoors have a great, vast diversity of the number of species that we see, and organic substrates or natural substrates have far less, but they still have a good amount. And I think we don't want to miss the presence of that and be able to understand that more. And so how do we increase our understanding of the types of microbes that might be present in these different substrate materials? Their function is extremely important. So there's a lot of genus and different species of fungi, bacteria, different microbes that might share similarities between ones that are beneficial and ones that are pathogenic. And so we need to understand the function, and then how can we leverage or promote those positive functions within indoor production systems. And then obviously preventing pathogenic competition. How can we understand the mix, the diversity, the abundance of microbes that we have so that we can limit any harm or any issues that pathogens may play and sort of outcompete those. Organic substrate materials are vast. We know the big ones to be peat, cocoa coir, wood fiber, and other wood materials. All of these come with their own microbiome and microbial diversity that, frankly, we don't know a lot about, and we haven't really had the tools to properly assess so that we can see how we can leverage those. There's many inert substrate materials, and the difference there, inert materials are not biologically active. They start in their production system completely sterile and void of a microbiome. But regardless of if we're talking about organic materials or inert materials, there is a way for us to incorporate microbiome regardless of the starting material. And I think what's really interesting when we dive into microbial diversity of materials specifically, we see very different presence and diversity. And so if we look at the left of bacteria across our three top ones, peat, cocoa coir, and wood fiber, we can see for bacterial presence, it's lower diversity for peat, and then cocoa coir, we would see generally higher bacterial presence. And then the opposite is sort of true when we go to fungi. So if we look at peat, greater fungal presence, lower bacterial presence, and cocoa coir, lower fungi and greater bacteria. And so peat and cocoa are actually the inverse, which I think is really interesting to be aware of. And then something like wood fiber we might see to be more consistent. The way we harvest and process these materials can certainly have an impact on the microbial diversity, but again, trying to get a better understanding of what is in these materials and how do we leverage those for positive enhancement within our production is extremely, extremely important. So in general, the driving question for me, for our group in this topic is really how do we leverage substrate microbial diversity for system resilience? So if we know that there is microbes present within our substrate, what are they? How do we leverage their function? How do we ensure we're getting positive microbes to outcompete and work to our good to increase growth and development of plants and ultimately yield and is what we're looking at. Regardless of the type of system, whether it's greenhouse, whether it's vertical, whether it's container, we have so many different types of systems that we employ, and this topic and trying to leverage this can be important regardless of the system that we're working with. So I will go ahead and pass this over to Paul, who's going to talk more about the work that they're doing at Concert Bio.
Thanks so much, Carl. I'm being overly modest as usual. So good morning / good afternoon, everyone. I'm Dr. Paul Rutten. I'm the founder and CEO of Concert Bio. And what we do at Concert Bio is really to try and enable this potential in the plant microbiome for growers in the CEA industry. So one of the things that I really like about this problem is that the potential for microbes in CEA is not just a woolly concept. It's something that you can actually measure. You can run experiments to measure what that potential is. And these experiments were, as far as I know, first done by a team at KU Leuven in Belgium. And essentially what you do is you start with something like peat, an organic substrate with lots of microbes in there, and you measure the yield of that. Then you switch to an inert substrate and you do quite a lot of sterilization. You don't kill all the microbes because the plant can't survive without at least some microbes there, but you get rid of a lot of them. And you see what that yield is. Now, there's a big drop from peat to an inert substrate. Part of that is because obviously you need to adjust the irrigation and other things when you switch to inert substrate, but not all of it. The next thing you can then do is to say, okay, let's put some of the chemicals that are in peat back into that inert substrate. There's lots of good organic compounds in peat, which are just not present in an inert substrate. And if you do that, you don't remove the microbes, you just put the chemicals in, we saw about an 18% improvement in yield. You can then say, okay, how much of it is the microbes that were in peat that are now no longer in this inert substrate? So you can do a separate experiment where you take just the microbes in peat, none of the chemicals, just the microbes, and you put that in the inert substrate. And when we did that, we saw this 57% improvement in the yield of the system. And actually the team in KU Leuven, when they did this, they did a similar experiment, but they were putting soil microbes into peat, and they saw a similar 70% improvement in yield, I think, is the highest they reported. So the potential is very measurable and very large. The challenge is just that about half the time when you do this, you introduce the wrong microbes into your CEA system, and there's a huge crash in yield. So what we really need is a small defined number of beneficial microbes that you can reliably add into these systems. So that's what we do at Concert Bio. We essentially develop these beneficial microbes that are tailored to CEA growers and CEA systems. And we do that based on what is now the world's largest database of CEA microbiome data, which we've been collecting for the last 3 years or so. So just to talk you through a little bit what the process is, how we collected some of the data that we use and that you also found in the white paper. We start by collecting samples of the substrate, the plant roots, also the fertigation solution in greenhouses and vertical farms. A lot of that is in these little tubes here. Any of you who have worked with us will recognize those. Those little tubes then come back to our lab in London. And what we do is we use metagenomic DNA sequencing. And the beauty of metagenomics is that unlike PCR or even PLFA, instead of looking for a limited list of microbes, with metagenomic sequencing, you can look for all the microbes in there, the good, the bad, and the ugly, as Marta likes to say. And you can also find microbes that you didn't even know you were looking for. Entirely unknown species will still be detected by metagenomic DNA sequencing. So what it gives us is this really nice complete map of the microbiome in hydroponic systems of both bacteria and the fungi. We've been at this for about 3 years or so, as I mentioned, and just to give you a sense of scale, we've got about 15,000 samples now from more than 40 growers spread across the US, Europe, the Middle East, and we recently even received a sample from the Caribbean. It's about 100 terabytes of data all told. To go back to the white paper, as Carl mentioned, one of the things that we learned from doing this is that there are certainly far more microbes in organic substrates like peat, but actually even peat pales in comparison to what you'd find in a soil sample. There are far more microbes in soil that plants expect to find around them and would normally harness around them than you would find in even an organic substrate like peat. What we can also do, if we dig into that data a little bit deeper, is to see where in a CEA system are these microbes. So if we start in the center there, if we look at the bacteria in a typical hydroponic system, the highest diversity is actually found in the nutrient solution. Bacteria are very good at living in liquid media. There's actually a slight drop in diversity on the roots. Plants will only let some of the microbes around them onto their roots. They have that ability to control them. And then there's actually an even smaller drop there into organic substrates. So you can see that as we get farther and farther away from the nutrient solution, the diversity of bacteria drops. What was very interesting was to see that with fungi, it's actually the opposite story. There's a very small diversity of fungi in the nutrient solution. A lot of these fungi just struggle to survive in the nutrient solution. Unfortunately, not things like Pythium and Phytophthora. There's slightly more fungi on the roots, and then there's quite a lot of fungi in the organic substrate. And what we've seen from our studies actually is that a lot of the fungi and the bacteria, something like a quarter in most systems, a quarter of the fungi and the bacteria that you find on the roots of the plants have actually come from the organic substrate. The plants have recruited these microbes from the organic substrate that they've been grown in. So substrate is a very important input for the plants to get access to the microbes that they need. What we've done with this data is to also try to make it more accessible to the growers that we work with so that they can start to get an understanding of what's going on in their microbiome. And ultimately, you can't improve what you can't measure. So we've tried to make it possible for growers to measure and respond to their microbiome in the same way that they can respond to temperature, pH, EC, and some of these other measurements that they are used to having control over. There's a lot of different things that we've learned from that, although we are far from done. There is still decades of research here, as Marta will tell you. But I'll give you an example of one thing that we've learned, which is we very often get asked by growers, what is the health of my microbiome? How healthy is it? How likely am I to have a disease outbreak? And we started with doing what we thought was the obvious thing to do, which is to look at what diseases are present in the system. So for a lot of lettuce growers and leafy growers, for example, it would be Pythium. And of course, with metagenomic sequencing, you can very quickly check not just for whether there's Pythium in there and what the level of Pythium is, we can even say this is the species of Pythium that you have in there. And there are more and less aggressive species of Pythium, which is one of the things that we've learned. However, what we saw is that most lettuce growers have some amount of Pythium in there. So actually checking for whether or not there is Pythium is not very helpful because almost everyone has some Pythium. We also saw over and over again that many people have a high level of Pythium, but no disease symptoms. And then there are some people who have quite a low level of Pythium and they still have obvious disease symptoms. So just looking at the level of a disease was not actually a very good predictor of how healthy the system was. What we then did is, thanks to metagenomics, we could take this a level further and we could start looking at the balance in the ecosystem. So because we detect all the microbes that are in there, we can look at what proportion of the ecosystem is being taken up by pathogens and what proportion of the ecosystem is being taken up by beneficial microbes or at least harmless microbes. And that ratio pretty consistently across different systems is about 80%. Normally about 80% of your microbes are beneficials and about 20% are pathogens. And what we saw is that very often when that ratio starts to drop, when you get more and more pathogens taking over the ecosystem, you are either in trouble or you are about to be in trouble. So this is the sort of thing that you can start to do with metagenomic sequencing that would be very hard to do with other techniques. So now we come on to the next stage, which is how do we use that data to actually find beneficial microbes in CEA systems? So we start with this complete map of the microbiome. We also get a lot of metadata from growers, whether it is their yield, temperature, humidity, pH, all of these other factors. What we can do with that is we can build ML models, and to cut a very long story short, what the models can do is they can find correlations between the microbes that are there and how well the plants are growing. And so what we can do is we can ask the models, okay, give me a list of microbes that seem to correlate with better yield. When you see more of these microbes, the yield tends to go up. We can then isolate those microbes. We can get our hands on them in our lab in London, and we can turn them into beneficial microbe products so that growers can intentionally add some of those microbes into their system. And this is our first ever product. This is Overture, which is a single species, one of these microbes that our models found from looking at all of this data. Just to give you a sense of what that physically looks like in the real world when people are applying it. Any of you who are growing leafy greens in CEA will be familiar with this. This is a large greenhouse in the UK that is using their spray bar to wet their seeds to kick off the germination. And what we've done with them and with many other customers is to dilute our microbes into that spray bar so that all the seeds are getting sprayed with the microbes. And the advantage of that is A, it's very practical and easy for the grower because they have this equipment already. But B, the nice thing is that we completely bypass the need to add our microbes to the fertigation solution. So it doesn't matter if you as a grower are using ozone or peroxide or UV filters or any other kind of sterilization system. We don't actually need our microbes to survive in the fertigation system because they're already sitting on the plants. And so it's a very efficient and very practical way of getting these microbes on there. So if we look at some of the results that we got, we ran about 14 trials on lettuce over the last 12 months or so. And we had 2 different products that we were sending to people, Overture A, which is one of these species, and Overture B, which is another species that the models found. And the punchline is growers were telling us that they had about a 9.9% improvement in yield on average. There were also trials done with CROs, independent CROs like Delphi, and with researchers. And these trials were run to anywhere where people would take these microbes. So we had trials in the UK, in the Netherlands, the EU, and the US. Now, unfortunately, it's science, it's not magic, so it doesn't work for everyone. There's about an 82% win rate. So about 82% of people who get the microbes will get an improvement in yield. The next question then for us was to say, well, the models obviously can't tell us why these microbes are working. So they're just finding correlations in the data. So we've then started looking around to see what is it that these microbes are doing. This is the genome of Overture A. And one of the things that we found in there, I won't say all the things that we found, but one of them is that there are 3 clusters for plant growth hormones. So these are genes that produce hormones that help plants to grow. And we think that is at least one of the mechanisms by which this microbe is helping these plants to grow. In general, what we see is that the microbes that we're finding are not microbes that help plants when there is a problem in the system. They are most helpful when you have a well-running system already and you're trying to get even more out of it. The better run the system is, the bigger the beneficial impact of the microbes is, generally. So just to round off on what the future looks like for us, hopefully coming to a CEA system near you. We're doing more product development now on vine crops. We've shown that our microbes can work on cucumbers and tomatoes. We're doing trials on peppers right now. We're going to launch Overture in the US this summer. We're planning to launch it in about the next 3 months or so for some of the customers who've already started it in the US. We'll be launching it in Europe, including the Netherlands, if any of you are based there, by the end of 2025. We're looking for more growers right now to help us do more trials and further optimize the use of Overture and collect those samples. And those samples are free of charge. So if you're interested in helping us do this and in getting access to the product, then please get in touch. My email is there, and there's a QR code if you want to find a little bit more information. And I will hand over to Marta now.
Yeah, thank you, Paul, for handing over to me. My name is Marta Stremińska. Everybody, good morning or good afternoon, depending on where you are. I'm a microbial ecologist at the Wageninegn University Business Unit, Greenhouse Horticulture. So if you think about the Netherlands, of course, yeah, that's the way to go when it comes to greenhouse horticulture, the start of it, and how we can make use of it to produce different products from vegetables to the ornamentals. If we look at the greenhouses in the Netherlands, we have different kinds of growing media we are using, from water systems, water-based systems, to inert growing media like stone wool or perlite, but also a lot of organic growing media like peat, coco coir, wood fiber, all different mixes of the three of these ones, or new mixes, peat-free mixes, which of course every single one has a different microbiome in it. And some of the cultivation, even in the greenhouses in the Netherlands, is still done in the soil, mostly for the ornamental plants. Of course, every single system, when we look at it, differs when it comes to the microorganisms present in the system. So we have a different complexity of the growing medium as a matrix, but also a different complexity as it comes to the microorganisms. And if we look at the water-based system and inert growing media, we know that there is a bacterial dominance in the microbiome, just like Kyle and Paul already mentioned. And we have, of course, organics, where a lot more complex organic matter is present, and a lot of more fungi are able to establish. Of course, in Wageningen, we're trying to figure out how to improve all the cultivation and improve the production and the quality and health of the plants. So we're trying also to make use of the microbes present in the system. That was a different story in the past. Very often growers wanted to disinfect everything, but we now see that there is a lot of options and a lot of good things that microbes can do in the system. Of course, we also know that the microbes that are present in the system are not the same microbes from the start to finish of the cultivation. If we talk about the cultivation of tomato, that takes around 10 months in the greenhouse in the Netherlands, we know that the microbes present at the beginning of the cultivation, or even the propagation phase, are different microbes that are actually present at the end of the cultivation, or during the process of flowering, of producing of the fruit. What has an effect on this diversity is, of course, the plant age. So what type of root exudate the plant is producing at a certain point in the cultivation, what plant species, and sometimes even a cultivar can have an effect. The type of growing medium, of course, have a very large effect if it's inert or organic, organic being also the source of microbes on its own. But we also know that nutrient solution microbiome can also be very important when it comes to the first establishment, for example, in inert growing media. And during the cultivation, usually growers do not use a lot of organic carbon, but also in the Netherlands, we are switching more and more towards the organic fertilizers. So they might contain some organic carbon, which can be also used by the microbes around the roots of the plants. So here I would like to give you an example of a project that we've done in the past when we compared the microbiome, bacterial microbiome, in the growing media during the cultivation of cucumber. We looked at two different growing media, coco coir slabs and stone wool slabs, and we also added biochar and spent mushroom substrate between the pot and the slab. So the organic carbon, when the roots grow into the slab, they can go through the biochar layer or through the layer of the spent mushroom substrate. And if you look at the right side of the slide, you can see a representation what kind of bacteria or what kind of genera of bacteria we could find in stone wool and coco coir. The colors are actually a different genera, so you can see in the first look at it that the stone wool and coco coir have different genera, but these genera also change over time, and this figure in the middle tells you that if we have different time points, the microbiome between the even if it's still the same crop like cucumber, the microbiome in stone wool and microbiome in coco coir, like this on the left is coco coir and on the right stone wool. But we see that they also change over time. So green and brown color, that are 2 time points taken, samples were taken month and 2 months after start of the cultivation. So we see also that the addition of biochar, of spent mushroom substrate, also can have an effect on the microbiome in these growing media. So that was our first indication that we actually can manipulate the microbiome in the growing media to obtain something tangible also for the growers, like, for example, disease suppression in the growing media, which also, unfortunately, is present also in the cultivation outside of the soil. So, of course, we also wanted to know what kind of microbes are present in the system. And when we look at it from the point of view of the plant cultivation, we talk about the good microbes like plant growth promoting bacteria and fungi. We talk about the bad ones, so the plant pathogens, so they will affect the plant itself, and possibly also the presence of what we call the ugly ones, so possible human pathogens. And we know, of course, that there are, if you look at the list of the microbes present in a system, some of them we don't really know what they're exactly doing at the moment. So we want to kind of figure out what they can do for us. And if we look at the problems that growers in the Netherlands face, is mostly just trying to get rid of the disease, of suppress the disease development. So we focus a lot, of course, on the pathogens, which can also very easily spread in growing systems outside of the soil, like oomycetes, Pythium, Phytophthora, which produce zoospores that can actively swim in the water system and in this way spread through the system. But we also look at the possibility of suppressing different fungal diseases like fusarium diseases, verticillium from the below ground, but also mildews, botrytis for the above ground in the system, which can also sometimes be transferred with the water system. And we also have a number of bacterial plant pathogens like Agrobacterium causing the what we call crazy roots or hairy roots disease in for example, stone wool mats, but also in an organic growing media. We have bacteria like Xanthomonas causing root rots, different kind of leaf diseases, but also some kind of a wilting, like, for example, Ralstonia. So these pathogens received a lot of interest from the growers in the past, but now we kind of try to persuade the growers, give them examples that there are more the beneficial microbes in the system than actually the pathogens. The thing is only how to use the beneficial ones to make sure that they actually do what we want them to do in the system. So basically, that starts with the disease suppression of the pathogen. The original definition of disease suppression comes from the soil environment, when we talk about that the soil is disease suppressive when the pathogen is present, but it's not causing any disease symptoms in the plants. And we kind of try to understand and try to do it within the growing systems in the greenhouses, out of the soil, inert growing media, but also organic growing media, and to enhance the disease suppression via addition of organic amendments like compost and amendments of chitin, but also by using the specific growing substrates. So mixes, for example, of coco coir with different things, where the specific beneficial microbes can grow, and at the end, of course, end up on the roots of the plant and protect the plant by antagonism towards the pathogens, by parasitism of the pathogens, or by the competition for the niche. So these are different ways how to steer the disease suppression in the growing medium. And some of the growing media can really be a source of beneficial microbes for the system. So this is one of the examples that we actually did in the past, also in one of the projects when we tried to manipulate the disease suppression in the growing medium by adding the spent mushroom substrate T. And we looked at the cucumber growth and inoculated also the cucumber pathogen, Fusarium. And if we look at the right figure here, we see the development on disease within the growing medium, which was not inoculated with the spent mushroom substrate D. And it's a lot quicker, and the disease is percentage is a lot higher when we don't use anything and we put Fusarium in the system, and when we use the spent mushroom T, we see that the disease is a lot lower when it comes to the percentage. And we also looked at the microbes present in both systems, as we see that through the addition of organic substance, but also probably microbes present in the spent mushroom tea, we can influence the microbiome of the system. So even in the growing medium, when we add an additional organic source, we can also manipulate the system towards the more disease suppression. So that's for us the one of the things that we've done in the past. We are, of course, still working on it. And just like Kyle and Paul mentioned, there are still a lot of unknowns. So we actually in the Netherlands, we look at it like we still have to persuade some growers that it's good that they have microbes, because they still think mostly about the pathogens and forget that there are loads of other microbes which are beneficial for the plant. And we can make use of them and use their potential to still increase the production and quality and disease suppression of the systems. But we, of course, would like to know still a little bit more, not only about which microbes are there, but what they are exactly doing in the system. So that's why we're looking also more and more into the functions of the specific activity of the microbes in different systems in greenhouse horticulture. So that's what I would like to share with you. So if you would like to know more, please do not hesitate to ask me. Thank you very much.
Thank you so much, Paul and Marta. I'm all about leaving enough time for questions, because that's truly where the magic happens. So if you have questions, please submit those through the Q&A, and we'll get to those in a few moments. I do have a couple questions that I'll kick us off with. We are so hyper-focused on technology development in CEA when it comes to systems, lighting, irrigation controls, all of that. And I feel like there's a huge opportunity for technology development in monitoring the microbiome. And I think that's probably a very complex task, but I'm curious if there's any thoughts from both of you on that. And maybe I'll start with Paul, because I know you all are maybe a little bit more closer to that in work on monitoring. But I could see a world where it would be super beneficial if a grower could sort of real time have a better assessment of how the microbiome changes, what's present in their system, as opposed to just maybe taking time points of a sample and then you're really just running that through a reference of if a pathogen is present, and not trying to flip it and focus on what are the positive microbes that are present, and maybe that's a better approach to addressing some issues. So curious both of your thoughts, but maybe start with you, Paul.
Yeah. So the reality is that as the technology that we have available to us right now just won't let us do metagenomic DNA sequencing in real time. It just, as an absolute minimum, the technology needs a couple of days really to be able to do that. And so we just don't have technology that gives us anything like that kind of insights into the system. You either have to wait at least a few days, or you have to take a huge hit and not really get much useful information about the microbiome that's there in these systems. So there's a trade-off there, and I think in general what we've seen is that the microbiome can evolve in these systems, but it also does not evolve overnight. So with the sort of turnaround time that we have, which if we're moving, then we can get a turnaround time of about 2 weeks or so. And that for most people is good enough that if something starts to go wrong, they still have some time to respond, especially for vine crops where the growing cycle is many months. But Marta, maybe you have some more thoughts on that.
Yeah, I think I can also say almost the same thing, that when it comes to really an in-depth information about the microbiome, the methods now are not like you can get the results within a couple of hours, but I think in a number of years they might evolve this way, because the whole PCR methods, metagenomics, all that evolves very quickly if you look at it. So I think we're going into that direction to get information very quickly, but there are, of course, methods based on DNA are just a tip of an iceberg when it comes to measuring microbiome in a system. There are also flow cytometry, things like that get the different kind of the information. But if there's something wrong going on in the system, sometimes you can really easily measure it, even with, for example, if you take your water sample and you see that at the end you measure, for example, ATP that's present in the cells of bacteria or fungi present in the system, and at some point you have a very low or actually very high, that you know at that point that something is happening. And of course the question is, you just need to follow up on this kind of measurements and figure it out how it works. But it's not these all microbial measurements are usually take time, and there's no one measurement that will tell you everything, unfortunately, at that point.
Absolutely. One question that came from our participants is, what evidence is there that applying commercial pre or probiotic mixes has lasting effects on the system or the plant microbiome? And so I think that question's kind of getting at, we can maybe inject something into our system and it's there, but will it last? How does it sort of set up shop within our system to ensure that maybe over time it's doing its job?
Can I maybe start addressing this question? We've seen that, of course, it depends on what are you injecting in a system. We can inject bacteria in a system or fungi that come from outside of the system. And the way if they're able to establish is also, are they able to establish in a given system? So they have to compete with other microbes probably already present in the system. So it really depends if they're able to do that or not. So it's very good an idea to find something that comes from the system and putting it back, because then the possibility of establishing is already higher. But we, of course, indeed can influence the microbiome of the system by addition of some kind of organics. We've done it with different things. And the very important thing is, of course, the how much do you put in of these organics? Because if you put too much, that is, of course, possibly also a problem in a system. So there's always this a lot of consideration. It's just not one recipe for all of the growing systems, all of the cultivation systems.
Yeah, just to add to that, I think in our experience, it really depends on what kind of intervention you make. As Marta was saying, if you add a prebiotic, some kind of nutrient that the microbes can eat, you can create a feeding frenzy, and that can have quite a big impact on the microbiome. In our case, with the trials that we run, we almost always do before and after metagenomic analysis when we add a single microbe to see what the impact is. And that is often very, very subtle, actually, especially if you're looking at something like a vine crop. A month later, it can be very hard to tell if anything has actually changed, because it's a single microbe amongst hundreds of microbes. And it's very rare for us to add a microbe, even a working microbe, and see that it takes up a big piece of the ecosystem. It's more like it fits into some small little niche somewhere that wasn't filled before, and that is enough for it to have that kind of effect. What we haven't really seen is any evidence of our microbes, for example, spreading and establishing themselves in the system. The nutrient solution for a lot of microbes is quite a harsh environment, and it is quite difficult to get something that really sticks around there. So you do have to keep applying it for lettuce at every sowing, for example.
This is a question, I think, specifically for you, Paul, but you mentioned existing products improving yield for already successful setups. Has there been research done on strains or products that could look to improve failing or struggling systems?
I mean, there's certainly research going on. I think in general, we focused on systems where a lot of the other questions have already been answered. The nutrient solution is optimal, the temperature is optimal, because the technology to deal with your temperature properly exists. The technology to deal with your pH properly exists, and so we're looking for what is the next big leap forward, assuming you can already solve those problems. The challenge is, of course, that in hydroponics, because a lot of those problems are inherently solved already, whereas in soil it can be very difficult to get the plant the right level of nitrogen, microbes that will help the plant with getting the right nitrogen uptake often struggle to work in hydroponic systems because the plant already has all the nitrogen that it needs.
Awesome. Another question for you again, Paul. Did you guys analyze oomycete diversity comparison between organic substrates? If yes, how was the comparison?
We definitely have done that analysis. I can't remember the results off the top of my head, but what I will say is that we don't often see oomycetes coming in on organic substrates. You see it here and there, but it's typically not a big source. Oomycetes really love to sit in the nutrient solution. What typically will happen is they will get into the system at some point, and then they will really establish themselves in the nutrient solution. And so the substrates are not often a major input. We do occasionally see seeds being an input for diseases. Substrates for oomycetes, not so much. I also see that there's a question about root diversity within the roots, are also on the rhizosphere. So that's a very good, very technical question. We generally look at just the rhizosphere, so everything on top of and inside the roots, but we are starting to do work now on what's inside the roots versus what's outside the roots. And we think that may be part of the reason that the effect of our microbes look subtle, and maybe more subtle than it is because a lot of the effect is happening inside the root, and it may be masked by the fact that you're not looking there specifically. I see another question about average increase. So the short answer to that is, for most people who are doing teen and baby leaf lettuce, one application is enough. If you're doing whole head lettuce and you're growing it for 6 to 8 weeks, and you do get a better improvement if you do a second application. And I will leave that last one to Marta.
Yeah, Marta, so there's a question for you on if you have an example of a plant pathogen present in a growing system and not causing disease. And I think, Paul, you could probably follow up after because you mentioned that as well. But
well, we have these kind of examples from our own research that if we manipulate the microbiome of the system, we can lower the disease incidence of Fusarium, for example, because we also add artificially pathogen into the system. But if the microbiome is changing through the manipulation that we do, that the plants are not getting sick. And the same we can also say for example, Pythium, because we know that Pythium is, of course, not very strong competitor with other microbes. So the total activity of the microbes probably will stop it from developing into the disease. With Fusarium, we see that it's more of a specific disease suppression, so specific microbes present in the system that work against Fusarium.
Yeah, I'll just add to that to say that we have seen many systems where there is a level of Pythium, a level of Phytophthora and other things, and there is no symptoms whatsoever. Just yes, no on whether the disease is there is not a good predictor of whether you will have an outbreak.
This might be maybe a follow-up question after, but a participant's asking if you can, any chance, you can share the data blinded to see more features, or if more features can be looked into, potential collaboration. I think there's something interesting in the data sets from Marta and Paul. I'm sure you all can reach out to Paul and Marta specifically to talk about those collaborations. I think that's probably a short answer.
Absolutely, no problem.
Another question is, these probiotics have a short shelf life. What's the what effect those environmental conditions have on engineering the microbiome?
I think I'll take this one. So this is still very early days for us in terms of figuring out what the environmental conditions in the system have on the effect of whether or not your products work. In general, most greenhouses have fairly similar temperature and pH and EC profile, and most vertical farms are also trying to get to about the same thing. So we often don't see environmental conditions having a big impact, but the variety of lettuce that you're growing, and certainly the substrate, can, we have seen, have quite a big impact on which microbes will work for different people.
Yeah, that's a good answer. We've talked a lot about plant pathogens. There's a question on human pathogens, which is equally important, if not more. So maybe you can start with this one, Marta. When considering the ugly for human pathogens, has any of your research seen a similarity between organic substrates and presence of a certain human pathogen? So does one material tend to have more of something than another, or is that maybe just a factor of it being introduced or handling, things like that?
When I look at the Dutch situation, we have the here organization that actually is taking care of what kind of growing media are being used in the greenhouses, also organic growing media, and these growing media have to undergo checks every now and then on the presence of possible human pathogens. So actually, I do not expect that there would be a lot of source from the growing medium of the possible human pathogens. The other story is, of course, the water. The water can be sourced from different places. It can be rainwater, but it can also be water coming from the groundwater or even in surface water. So there might be some contaminants, but I would not expect that possible human pathogens would come from the growing media.
Another question I have in your presentation, Marta, so you talk about harnessing the potential of microbes as opposed to disinfecting, and I think that's such an important question because we as humans often like to just find the problem and eliminate that, and sometimes that comes with eliminating everything for just risk mitigation, and I think we know, and we've talked through, that's not always the best strategy. But how do you practically do that for a grower who says, this is really interesting, I'm compelled, and I want to harness the potential of the microbiome, but where do you start? Because the microbiome is very complex, and I kind of feel like we haven't focused enough on this topic just because it is so complex that it's easier to just kind of brush aside and focus on something that we can control a little bit more.
Yeah, I think it's maybe also people have a different idea what all the disinfection methods do, because we have research showing that even if you disinfect your water supply, so with UV light, with maybe hydrogen peroxide, of chlorine dioxide, and so on, there are still a lot of microbes present in the growing medium and around the roots. And these treatments in the water, they're of course needed to get rid of the pathogen in the water, so you will not recirculate, for example, pathogen in your systems. But you have to be careful when it comes to the concentration of different oxidants, for example, that you use, that it does not affect in the end also the microbes around the roots. But there are still a lot of microbes around the roots that are not being affected by the disinfection. Disinfection is also very important, what kind of disinfection you want to do, because we also know that if you use oxidants, you can actually cause the development of of pathogens, for example, bacteria pathogens that are more and more resistant to a higher concentration of the oxidant, like hydrogen peroxide. We see that, for example, in Agrobacterium, that's a bacteria that's causing these crazy root symptoms in plants, and they at the moment can live through 600 ppm of hydrogen peroxide. So it's quite a lot, because 600 ppm hydrogen peroxide is not very good for the roots of the plants growing in the same system. So yeah, this is always getting rid of the pathogen. It's very important. Just make sure that the system is pathogen free, but it doesn't have to be sterile to get to that point.
Absolutely.
Yeah, I can very much echo that. One of the things that most surprised us when we got started was how largely ineffective a lot of these sterilization systems are compared to what people expect. They kill a lot less things than people think they are killing.
Yeah, that's a good point. Another question came through: as this technology grows and there's more niche microbes for plant variants that are used or applied, any thoughts on how we can address the Nagoya protocol for microbes used in commercial application? Paul, you might be more familiar with that, but I'm not sure if, yeah.
Yeah, it's something we've thought about. It's not something we've had to deal with much so far. A lot of the microbes are things that we are isolating from the systems themselves, whether that be in the US or in Europe or the UK. But yes, indeed, at some point in the future, that may become a consideration, because a lot of the microbes that we use, all of the microbes that we use, are naturally occurring microbes. We haven't genetically engineered them in any way, so there is a country of origin for them.
Yeah. Another question I have, so, and you mentioned this, Paul, but it seems like a lot of the available products on the market kind of focus on one microbe or one strain, and I'm sure it's easier to sort of work with one than a cocktail of many, but since we're talking about a microbiome and diversity, is it beneficial to move towards sort of a broader mix, or it's generally just focus on one, and that one, should have the greatest impact?
So we look at it really as a trade-off. If you add more species of microbes, then the potential effect you can have inherently will get bigger, assuming that those microbes you're adding together get on with each other, which is often challenging, and that they don't get in each other's way, and that you can produce them together. For us, as a fairly early stage company, we think about, okay, how big of an improvement can we get with just a single microbe, and how much bigger is the improvement if we put multiple microbes together, and then how much harder is it to produce those and apply those and get them working for people. And if it is only slightly better but much harder to make a cocktail work, then we don't do a cocktail. We start with just a single microbe, which is what we've done.
Right. I'm going to finish off the last question for you, Marta, and then we can wrap up. But you talked about plant growth promoting bacteria and fungi, and I think we as humans always like to cling to the negative things, and so we focus on Pythium, we focus on Phytophthora. What are some of the positive plant growth promoting bacteria, fungi, that our audience can just be aware of, that we see in systems?
Yeah, for example, very well-known Pseudomonas fluorescens. That's a bacteria that we often hear. That's a plant growth promoting bacteria, and we even see it in the growing system with stonewool and organic growing media developing around the roots on its own, from natural way, without addition of these microbes. So the plant growth promoting bacteria and fungi, I would think that we kind of, with the move from the soil into the soilless growing systems, we kind of forgot about them, because we think that the production is the maximum what we can get. But I think there's still a lot of to get out of the beneficials. Like they can produce plant growth hormones, so we can steer the plant production more in a way that we want it to go. They can also really influence the nutrient availability, and in the past it was not such a big issue, because there was a lot of mineral nutrients present, and they are being used also in greenhouse horticulture. But we are switching again into the circular horticulture, so reusing a lot of organic organics, also fertilizers, organic sources of fertilizer, and these need to be mineralized by the microbes before they are really able to be used by the plant. So yeah, these kind of things, I think the microbes are really going to play a very important role in the future, even if we have a lot of control, like we would like to think about it.
Perfect. Thank you. Well, that concludes our time. I will just address the last comment. I know Paul and Concert Bio have collected a lot of those environmental factors in their data, and I think they sort of isolated that it doesn't have as much of an impact as some other elements, but I think it's that analysis of that data would be nice to see in terms of what factors have been addressed and what can have an influence. Again, thank you so much for joining this webinar. It's a really exciting topic and one that we need to focus on more. And I know there's a lot of good research going on. Wageninegn, Jiffy's a part of some research Ohio State's doing on this in the US. And so there's a lot of different research efforts trying to better understand the microbiome and the positive influence it can have, because it's a complex situation and takes a lot of effort to figure out. So thank you for attending. Again, this is recorded. We will provide that. And also just follow up to give you a chance to download that white paper. Please reach out if you have any other questions. Enjoy the rest of your day and have a wonderful weekend.
Yeah. Thank you very much. Have a nice weekend.